Okay, so my lab is interested in a fundamental question: how cells divide, how cells proliferate, and really this is a question with relevance in many diseases, including cancer, but also many others, right? So the critical question in our lab is, how most of the signaling pathways trigger the cell division cycle, how cells are convinced that they need to proliferate, and obviously a major question here is if you are able to discriminate between normal cells and tumor cells, and which are the critical players that are responsible for that in tumors, and again, this might apply to many other diseases. So, this question was kind of solved many years ago with several family of proteins, including cyclin-dependent kinases, working in yeast and other models, right? So these are proteins which are critical to, again, go from a cell which is quiescent, not dividing, to the cell division cycle. So the problem in mammals is that we have 20 different cyclin-dependent kinases. And again, there are many other protein families, but I think this is one of the main engines of the cell division cycle. And obviously, this is a problem for, try to answer the question, which is the critical CDK responsible for this disease or this disease. And right now, there are, many CDK inhibitors, and there are many diseases, including, cardiovascular disease, Alzheimer, obviously cancer, in which specific CDKs are important for the disease and might be good therapeutic targets. So the problem with the CDKs is not only the kinases themselves, but the name comes from cyclin-dependent kinase. So these are kinases that are inactive, and they require an additional subunit, which is known as cyclin, to be active. And this is a cartoon of the family of cyclins. So we have like 20 CDKs, we have 35 cyclins, and you need to combine one with the other to have an activity, right? So that makes everything very, very complex for disease. So this is a 3D structure, and really now we have several inhibitors that go into the catalytic pocket. But again, this needs to be specific for a cyclin and a CDK. And again, if I put everything together, to the left you have the CDKs, to the right you have the cyclins. And many years ago I started to put a line from one CDK to the cyclin, based on the literature. And this is some of the lines I was, drawing. And I have to tell you, I stopped doing that in 2005, like, I don't know, 17 years ago, because this was very, very complicated, right? And obviously, if I continue to do that, there are many, many, many possibilities. So now the question is, how are you able to propose one of these guys as a cancer target? So one possibility is, again, starting from the preclinical studies, in vitro studies, you might eliminate every gene, in cell lines, right? So this are some data from the map repository. So this is a repository where you have 1,000 cell lines, and you knock out, so you eliminate a specific gene in any of the 1,000 cell lines, right? So now you can ask, which genes are important or not. So this is to the right, you have a cartoon. This is our data we are still generating, but this is a cartoon in which you see the dependency index. That means that if you eliminate this specific gene, cells do not proliferate. So the decrease in proliferation is very high, right? That means that, for instance, CDK1 is very important for most of the cell lines. If you eliminate CDK1, most of the cell lines, I mean, these are cancer cell lines, but this also applies to normal cell lines, do not proliferate, right? So that means that probably CDK1 is toxic, is too essential, and that might apply also for CDK7 and CDK9 here. But there are others in which you can see that, well, maybe depending on the cell line, you might have an effect. I have to tell you that every spot in here is the average for a specific tumor type. For instance, if I tell you, which are the tumor types in which CDK4, again, one of the family members, is important. So if you eliminate CDK4, these tumor cells do not proliferate. So number one, the most dramatic effect is, for instance, bone, and this is with cyclin D1, one of the cyclins. Second one is prostate, third one is breast. They are very similar. So that means that specifically these tumor types are very sensitive to CDK4, but maybe not other cell types, right? Again, this is a kind of preclinical assay, but actually it's very informative because now, again, stemming from this example with CDK4, actually CDK4 inhibitors having approved for breast cancer, right? So this was predicted from the analysis with the cell lines, and now we have like 5 different CDK4 inhibitors, 3 of them approved in Europe, that specifically inhibit 2 members of the family, which are CDK4 and CDK6. And they are only approved for hormone-positive breast cancer, right? Why is that? Again, if you imagine that, one cell needs to divide in two cells, the machinery is very similar, but the trigger, the enzyme that determines that this happens might be different in, pancreatic beta cells, in lung cancer cells, or in breast cancer cells. So it happened that CDK4 specifically is downstream of the estrogen signaling pathway, and therefore is very important for breast cancer. No? So now the question is, what are the others? So how are we able to map all the others? Obviously, we have some preclinical tools, but we also have a lot of pharmas interested on that. So this is an update of, this was April this year, more than 500 clinical trials with CDK inhibitors, which is huge. It's not only cancer, many diseases. And, as you see here, there are inhibitors for many family members, right? So these families typically divide into the cell cycle proteins, which are responsible for the cells to divide, and also transcriptional CDKs, because they phosphorylate many different transcriptional regulators. So if you realize about this cartoon, most of the family is kind of taking, most of the companies are interested in one of the other family members, but there is a group, which is this CDK 16, 17, 18, 14, 15, which almost no papers, no pharma companies, no drugs. So a few years ago, we decided to start working on that. So we generated CRISPR reagents for testing these family members. And I have only 2 slides with the results. So this is a CRISPR, meaning elimination of specific genes. In this case, this is a liver cancer cell line. And as you see, if you eliminate only CDK14 or 15 or 16, which are in the top, you see no effect. But if you eliminate 2 of them, for instance, 14 and 16, or 16 and 17, or 18, you do have a strong effect, right? This is a general summary or a general message from the family, that typically, because you have so many family members, they overlap, they compensate. So in some cases, you need to eliminate 2 or 3 maybe, right? So if you pay attention to the color information assay, again, if you eliminate 1 of them, no effect. If you eliminate 2 of them, 16, 18, is very dramatic, right? So we also demonstrate that in vivo. So for that, in the past, we generated mouse models, so meaning conditional knockouts or knockouts for, most of the cell cycle family members, CDK1, CDK2, 3, 4, 6 in the past. So in the last few years, we generated 5 new knockouts for each of the new family member, right? So we already have all these animals running. And we want to generate double and triple knockouts. But I'm going to show you just one example, which is what is the effect in liver cancer. So we took mice, we induced liver tumors over injecting MYC oncogene and beta-catenin, and now we test whether eliminating 1 or 2 of the CDKs has any effect, right? And this is the result. So to the left, you have these liver cancers induced by MYC and beta-catenin in a wild-type animal. So in the quantification, you have the ratio in the body weight, or sorry, in the liver weight, or the number of tumors. And to the right, you have the effect of eliminating CDK16 and CDK18. I have to tell you that the elimination of the genes with the genetic knockouts was a therapeutic effect. So once the tumor was identified and quantified by imaging techniques, right? It's not germline, it's not preventive, it's a therapeutic effect of eliminating CDK16 and 18. Obviously, now we are testing other clinical scenarios, but the summary from here is, well, we started working on that, with the idea that cell proliferation and the cell division cycle is universal. At the end, all the cells have kind of the same machinery to divide, but still we don't understand, how this is different in a breast cancer, or in a hormone-positive breast cancer, or in a triple-negative breast cancer, and many others. Number 2, we already have some positive data, specifically and only with CDK4 inhibitors and only in hormone-positive breast cancer. And obviously now there are many reasons for continue working on that. Number 1, most of the metastatic tumors relapse after CDK4 inhibitors. And so we are investigating biomarkers and mechanisms of resistance. Also, we have novel targets, the ones I already presented here, and new therapeutic opportunities. And still, we need to understand which are the clinical scenarios that might be sensitive for this disease. So with that, I will stop. So this is my group at the VHIO, which is working now. So we are taking people interested in joining us. Thank you.